Once again, I'm meeting you after a long time. Now, the lesson is only for the pure science students, not for bio students. Some of the parts left in chapter 6, evolution, that is pure science. What is the chapter? Isolating mechanisms. What is isolation? So, isolation normally resulted in the formation of some subspecies or subpopulations. And these are all the barriers. The isolating mechanisms are the barriers responsible for the formation of new species. What is isolation? Isolation is a separation of a single population into many subpopulations. So that what will happen? The genetic integrity of the subpopulations can't be maintained. Okay. So isolation is a separation of a single population into many subpopulations so that now the genetic integrity of subpopulations can be maintained. So now you know that one closely related species living together in the same area do not breed together. They are not breeding. What's the reason for that one? So the barriers, the isolating barriers prevent them from what is called breeding. So closely related species living in the same area do not breed together. What is the reason? They are prevented from interbreeding by means of what we have isolating barriers. The isolating barriers may be geographical isolation or reproductive isolation. So we have mainly isolating barriers or geographical isolation and then reproductive isolation. We'll talk about. So these two isolating mechanisms are responsible for preventing what is called interbreeding between the individuals of the same species. So what will happen? A single population is being divided into many subpopulations. They maintain the integrity, what is called the genetic integrity, because they are being separated by means of barriers. And these barriers that prevent them from interbreeding are called isolating barriers. What is an isolating barrier? An isolating barrier is an evolved character of the two species. Evolved character, not already present, the character that is evolved. The evolved character of the two species that prevent them from interbreeding. And that is called as an isolating barrier. Is an evolved character of the two species that prevent them from interbreeding. In due course of time, those characters are evolved. Now, the subspecies resulted because of what is called geographical barriers or reproductive barriers. Now, the new subspecies, for example, if you are taking crow, there are three different types of crow. There is one Indian crow, another one what we have the Sri Lankan crow, another one what we have that is Myanmar crow. So, what we have? Different types, splendens, Corvus splendens, that is the scientific name of crow. We have three different subspecies. Corvus splendens insolens in India, Corvus that is splendens protegatus in what we have Sri Lanka, like that we have. So the isolating mechanisms result in the formation of subspecies. Now we have several types of isolating barriers or mechanisms that prevent interbreeding. The most important distinction is number one, prezygotic isolation. The isolation that occurs before fertilization, preventing the individuals from coming into contact. Preventing the individuals from coming into contact. Now, postzygotic isolation, the isolating mechanisms that normally prevent, that is what we have interbreeding, acting after fertilization. So prezygotic, the one which occurs before mating, before what is called the zygote formation and before fertilization. The second one is acting after fertilization. Though fertilization occurs, because of certain factors we have, that is the individual will not develop, or they are sterile, or actually they are present but unable to reproduce, etc. So anyway, two major isolating mechanisms as per the book. We have prezygotic and postzygotic isolation mechanism. These two are coming under what is called reproductive isolation. These two are coming under reproductive isolation methods. reproductive isolation. Another major type is a geographical isolation. That is, the individuals are separated from one another by means of geographical barriers in the form of rivers, in the form of mountains, in the form of valleys, or in the form of ocean. So we have just different types of rock found in India, South India, and Sri Lanka. These two frogs are separated by means of water body, Gulf of Mannar. So anyway, we have two different types of reproductive isolating mechanisms. One, geographical isolation, that is not mentioned here. Species formation by means of geographical barriers. Then reproductive isolation, the reproductive mechanisms which will prevent interbreeding, so that what will happen, that is no interbreeding occurs. So they are called isolating barriers. We'll see one by one the prezygotic isolation mechanisms and postzygotic isolation mechanisms. So there are two different types of reproductive isolation methods. One is operating before what is called reproduction, preventing the two different species from coming into contact. Another one is operating after fertilization, that is after mating process, that is called postzygotic fertilization. Now let's take what I mean by prezygotic mechanism. Now these mechanisms normally includes those which prevent two species from coming into contact. So coming into contact. So preventing two different species from coming into contact though they are present in the same area. Now this includes a number of mechanisms. One, that is ecological isolation or what we have habitat isolation. We'll discuss. Seasonal isolation. Then sexual or ethological isolation or what we have behavioral isolation. This is not sexual, behavioral. Okay, so behavioral isolation. Sexual behavior, behavioral isolation. Ethology means study of behavior. That's why it's called ethological isolation. Then morphological and mechanical isolation. The differences in external genitalia structure between two different species not allowed to mate. Then physiological isolation, some physiological barriers. Then cytological isolation, difference in number of chromosomes between two different species or the structural differences between the chromosomes of two different species. Okay, let's see one by one. Now the first prezygotic isolating mechanism, ecological or habitat isolation. So the members of the same population may be separated from one another. Members of the same population may be separated from one another by differences in their habit, so that they may not interbreed. They maintain their respective species identities. Okay, so the members of the same population may be separated from one another by differences in their habitat that prevents what is called from interbreeding. So these species maintain their species, respective species, what is called just representations. Now, two species. Number one, that is with respect to the frog. One species, Rana areolata. This is one species. Another species, Rana grylio. These are the two examples. They are living together, just normally belong to the same genus but different species. Now, what about the nature of this Rana areolata? It occupies burrows. Those burrows are made by mammals and tortoises. Living in burrows made by what is called actually are done by mammals and tortoises. So they are just normally taking rest during the day and breeds in grassy, shallow ponds. Breeding occurs in grassy, shallow ponds. Ponds reaching grasses. That's called grassy, shallow ponds. The depth is low. Suppose you're taking another species where Rana grylio, whereas Rana grylio, it breeds in deep waters. So one breeds in shallow waters, another one breeds in deep waters, present in the same habitat, but they maintain their species, what is called actually identities. So occupying what is called just in the same habitat but at different places. So one is occupying what is called actually there is deep waters, another one is occupying occupying the shallow water ponds. So the breeding occurs in two different places because of the differences in their habitat. So due to the difference in their habitat, the two species are able to maintain their respective species identities. They do not interbreed because they are living in different habitats. One in shallow ponds, another one in deep ponds, not in the same. This I am telling that one in stratification shallow and deep. But here we are talking about different habitats. So one habitat shallow water, another habitat deep waters. One is breeding in shallow waters, another one is breeding in deep waters. That's why the two species may not interbreed because of differences in their habitat. Now the second type of prezygotic isolating mechanism, seasonal isolation. Now normally the breeding season is different in different animals that prevent what we call interbreeding. That's why the species actually identities are maintained. So in this type of isolation, differences in the breeding season prevents interbreeding between two different species. One species is breeding in one time in one season, another one is actually breeding in another time. This type of isolation is also called as temporal isolation. There is another name for this one, seasonal isolation or temporal isolation mechanisms. So reproduction in different seasons prevent interbreeding between two different species. Here is given example, the toad. The example, two different types of toad given. One is Bufo americanus. Now this one breeds much early when, that is in the spring season. So it breeds much early in the spring season. Whereas another toad, Bufo fogleri, that breeds very late in the season, very late. So this is in the early spring and this one in the late spring. That's why we have these two species do not interbreed. So they are able to maintain their species identity. How? Because of the differences in their what is called breeding season. They have different breeding seasons, so they do not interbreed because the reproductive organs are not ready at a particular season for two different species. They are ready only at two different seasons, thereby preventing interbreeding between two different species. So that's about seasonal isolation. Because of the differences in breeding season, ultimately what will happen? The interbreeding is prevented. Now the third method of prezygotic mechanism, ethological or sexual isolation. It is also called by another name behavioral isolation. Ethology, the word refers to the study of behavior. Now this prevents mating between two different species due to differences in their sexual behavior. The differences in sexual behavior that prevents what is called mating or interbreeding. So the species are not separated from one another either by time or space. They are present in the same place, in the same time, but they are prevented from mating. What is it due to? Mainly the mating cells, that is the gametes, the germ cells, sperms and eggs. So the mating cells of the two closely related species of frogs are different. We are taking the example of two species of frogs. The mating cells or the gametes or the sperms or the eggs of these two species are different, which prevent interbreeding. So mating cells are different in that what is called actually the moment or the nature or capacity, etc. So two species of tree frog, what is called Hyla versicolor and Hyla femoralis. This is a tree frog and that is adapted to live on trees. They have the ability to glide from one tree to another also. Hyla versicolor is a grey tree frog, grey colored tree frog, and another one Hyla femoralis. This is actually called as pine wood tree frog, found living on pine trees, hence the name. So in these two, though they belong to the same genus, their gametes are different in their functions or in their structure, etc. So what is happening? There is no mating occurs between the gametes of two different closely related species. Now the fourth type of prezygotic mechanisms, morphological or mechanical isolation. This is isolation, that is due to differences in the external genitalia. One species external genitalia is not compatible or not matchable with the external genitalia of another species. So we have differences in the external genitalia that is seen in two different species. Now, the size difference, the external genitalia, for example, the penis, and also the depth of what is called the vagina in different animal groups, preventing interbreeding. So, the size differences in external genitalia between two toad species, one before Corsicus, this is one that is a toad, and another species before Veliceps. So they are different in their size. The differences in external genitalia prevent interbreeding. That's why it's called mechanical isolation. The morphological structures related to the external genitalia, so morphology, the structural aspects. In relation to the external genitalia, they are different in their size, etc., between two different species. So these external genital organs are not compatible, not matchable between two different species, thereby preventing interbreeding. The best example, we have toad species before Corsicus, another one before Veliceps. They have just, and we see that when both are belonging to the same genus but different species, that external genital organ is different, not matchable. That prevents interbreeding. Now the next one, fifth one, cytological isolation. So fertilization is prevented between two different species due to difference in the number of chromosomes or difference in the structure of the chromosomes. Here in a book it is given only difference in the number. So fertilization does not take place due to differences in chromosome numbers. This is because of that one. No just fertilization occurs between two different species gametes. Okay, so normally in the case of what we have the frog, one is the bullfrog, another one gopher frog, bullfrog and gopher frog. In these two cases, we have the chromosome number is only 26, though it is given due to differences in chromosome, but in these two species, we have the number of chromosomes 26. But if you are taking the chromosome structure between 7 and 10, that is, we have between these two species that are different in their structural aspects that prevent them interbreeding, not allowing actually to fuse, not allowing what is called the fertilization process. Though these two frogs have same number of chromosomes 26, but as per the book different number. But what is happening? Differences occur in structural aspects between some chromosomes. That results in what is called preventing the fertilization process, leading to the absence of interbreeding. Now the sixth prezygotic isolating mechanism, physiological isolation. So in this case, mating occurs between two different species, but fertilization is prevented because of certain mechanical or physiological factors. For example, two species of Drosophila, the fruit fly. So the spores of Drosophila virilis, one species, when introduced into the female body and stored in the sperm receptacle, it survives only for a day one. So the sperms of Drosophila virilis survive only for about a day when introduced into the sperm receptacle of the female Drosophila, or what is called Drosophila americana. This is another species. But the sperm of the same species, Drosophila americana, when introduced into the sperm receptacle of what is called the female, they survive for a longer time. So, the sperms from the same species are accepted by the species and survive for a long time. This is because of certain factors favoring them, that is, in the female body of the same species. But the sperm from different species, when entered into the what is called the female reproductive tract, that conditions, the physiological factors present in the female reproductive tract of a different species prevent the sperm to survive for a longer time, thereby preventing what is called interbreeding. So the physiological barriers formed in the female body of one species prevent the survival of the sperm received from different species. But from the same species, it is living for a long while and fertilization is normally occurred. So this is another factor, the physiological isolation. So this is the last method that is coming under prezygotic mechanisms operating before fertilization, before the formation of zygote. Now we have to go for the next one, postzygotic mechanism, which includes those that normally occur after fertilization. We have three factors or three types. Let's see. Now the second method of what is called just isolating mechanism, the postzygotic mechanisms. These mechanisms are operative only after fertilization process. Fertilization occurs. Even though fertilization occurs, either the individual are inviable, that means dies, or the resultant individual are sterile, or you have F1 individuals are fertile and F2 individuals are sterile, like that. So postzygotic mechanisms are those which act after fertilization. See that one. That's why it's called postzygotic mechanisms. Natural selection normally favors those individuals having prezygotic mechanisms than those with postzygotic. So the individuals with the prezygotic mechanisms are favored and selected by natural selection, and individuals with the postzygotic mechanisms are eliminated. They are not surviving. So they are not having what's called the fittest organisms, the fittest nature of organisms. So they are being eliminated. Now these postzygotic mechanisms include one, hybrid inviability. Hybrids are formed but they die before reaching the maturity. The second one, hybrid sterility. Hybrids are formed but they are unable to reproduce. That is a F1 hybrids. A hybrid breakdown. The F1 hybrids are fertile. When two F1 hybrids are self-fertilized or self-mated, then we have F2 generation is resulted. They are, that is normally sterile. Now let's take hybrid inviability. In this type, what is happening? Sperm enters the egg, number one. Fertilization also occurs. And the embryo develops into the adult. All these stages are taking place, but it does not survive. The meaning for that one, it dies. It dies before reaching maturity. Even in the embryonic stage itself, the embryo dies. So, no survival of the offspring in this form. This is inviable. Okay. So, it is common in the case of some fishes, in the case of frogs and beetles. Even though fertilization occurs in these individuals, that is between two different species of these individuals. Now, due to genetic incompatibility, what is incompatibility? Not matchable. Compatible, matchable. The genotypes are not matchable. The chromosomes are not matchable. The genes are not matchable. That is called incompatibility, genetic incompatibility. So they do not leave any surviving offspring. No representative resulted from these. Actually, the fertilization process. The hybrids are formed but inviable, unable to survive. This is because of genetic incompatibility. The genes, the chromosomes, and their numbers, the structures are not same between these two different species. That's called genetic incompatibility. That's about hybrid inviability. We have to go for hybrid sterility. Now the second type of postzygotic mechanism, what is called hybrid sterility. Now in this type, what is happening? Hybrids are formed because of what is called interspecific process, but they are sterile. So we can make actually a cross between two different species belong to the same genera, but the resultant hybrid, the F1 hybrid, is normally sterile. What is it due to? This is because of unpairing of chromosomes. The chromosomes are of different types, so they do not pair, and also they do not segregate during meiosis, during the formation of gametes. So the sterility is due to the failure of chromosomes to segregate, that is normally during meiosis. That means the unpaired chromosomes, the homologous pairs are of not same type, and also they fail to separate during meiosis that resulted in sterility. Now here is an example, a cross between a donkey and a horse. Now the first cross is between what is called a male donkey and a female horse mare. So that is equus asinus. That is a binomial of donkey. And this one horse equus caballus. Both belong to same genus but belonging different species. Now the F1 hybrid is formed. That F1 hybrid is mule. That is sterile. This is called hybrid sterility. When reciprocal cross is effected, that is, we are taking the female donkey crossing with what is called the male horse. This is called reciprocal cross. The resultant product is F1 hybrid, but it is called Hindi, not mule. So cross between female, that is what is called the horse, and male donkey, that is mule. The cross between female donkey and male horse resulted in F1 hybrid, which is sterile, and that is called Hindi. This is the best example for hybrid sterility. F1 hybrids are formed, but they are unable to reproduce because of the failure of the chromosome to separate or segregate during meiosis. The last method of postzygotic mechanisms, what is called hybrid breakdown. When cross is effected between two parental combinations, that results in F1 hybrid. The F1 hybrid is fertile. When self-mating occurs between F1 hybrids, we have received what is called F2. The F2 generation is sterile. For example, if you are making a cross between male lion and female tiger, Panthera leo and Panthera tigris, the product in F1 hybrid is called liger. By taking the first two letters of what is called actually lion, the male, we are giving importance, and then the three letters of the last, that is tiger. So that results in liger. When two ligers are crossed, we are getting F2 individuals, but the F2 individuals are sterile. This is called hybrid breakdown. F1 fertile, F2 sterile. But in hybrid sterility, F1 itself is sterile. When reciprocal cross is effected between a female lion and a male tiger, then we are getting what is called trigone. The product is called trigone. That is fertile. When two digons are actually self-crossed, then what will happen? We will get F2 individuals. The F2 individuals are sterile. This is called hybrid breakdown. So these are the different mechanisms, what we can say isolating mechanisms, isolating barriers which prevent interspecific crosses to maintain what is called specific identities. Okay, so I'll take the remaining part of that evolution in the next class. Speciation, extinction, etc. Thank you.